In this video, we will discuss how the vaccines that are made up of Adeno Associated virus helps in providing information about the antigens to the immune cells or the T cells. So there are 5 different ways that are presented in this figure. We will go through all of them one by one. So we will start with this first possibilities, that is direct presentation. So in this case, the vaccines that are made up of adeno associated virus vector is injected into the muscle cells, and those adeno associated virus directly infect the APC or antigen presenting cells, and these antigen presenting cells then move to this draining lymph node where it will provide the informations to the T cells through the major histocompatibility complex. So this way of providing antigen informations to the immune cells is known as direct presentation. Uh, However, it is not always the case. There are sometimes the different other ways also occurs through which antigen informations are provided to the immune cells. So next one is this cross presentation. So in this case, the adeno-associated virus infect the muscle cells, and the muscle cells through this phagocytosis by the antigen presenting cells got the informations about the antigens that are enveloped into this adeno-associated virus, and thereafter these antigen presenting cells move into the draining lymph node, and then it will provide information to the immune cells through this major histocompatibility complex. So this is known as cross presentation. So in this case, the muscle cells got infected with the adeno-associated virus, and thereafter those through this phagocytosis, the antigen information is being transferred to the antigen presenting cells, and then it will transfer to the T cells in the draining lymph node. In some case, what will happen is that it's not the professional or the mature antigen presenting cells provide informations. The mature antigen presenting cells got infected with the adeno-associated virus, and thereafter these mature antigen presenting cells provide informations to the naive antigen presenting cells, and then it will move into the draining lymph node to provide informations to the T cells. So the difference between the cross-dressing and the direct presentation is that the different antigen presenting virus provide informations to the T cells in the draining lymph node, not the virus that has infected with the antigen presenting cells. Whereas in the case of direct presentation, the virus that are infected with the antigen presenting cells provide informations to the T cells in the draining lymph node. Whereas in the case of cross dressing, the virus that is the APC that is infected with the antigen presenting cells that is infected with the adeno associated virus provide information to the naive antigen presenting cells that will then move those antigen informations to the T cells in the draining lymph node. The next one is this local presentation. So in this case, what will happen is that the antigen presenting cells, it is similar to the direct presentation. Only difference is that antigen presenting cells does not have to move to the draining lymph node. It can directly provide information to the T cells at the site of infections, or the T cells are present at the site of infections. So the antigen presenting cells that are infected with this adeno-associated virus provides information to the T cells at the site of infections through the major histocompatibility complex. And the other ways similar to the cross presentation is that the adeno associated virus infect the muscle cells, and through the phagocytosis those antigen information that are present in the adeno associated virus will be transferred to the antigen presenting cells, and then it will provide those information to the T cells in the local environment or at the vicinity of the vaccine injection. So this is similar to the case 1 and case 2. Only difference is that the T cells are present at the site of vaccine injections. Last one is this local presentation without antigen presenting cells. So this is similar to the case. It is not exactly similar to any of the case 100%, but this is more direct contact informations, or there is the chances of antigen formations against the virus is more in this case. So what will happen is that the infected muscle cells, adeno associated virus got muscle cells got infected with this adeno associated virus at the site of injection, and thereafter those muscle cells provides directly information to the T cells without requiring any intermediate of that is antigen presenting cells. So this is known as local presentation without antigen presenting cells. So in this case, since there is no intermediate, the time required for antigen formations against the virus, against the antigen informations that are enveloped in this adeno associated vector will be fast compared to the all other case. So these are the different ways by which the adeno associated virus provides informations to the immune cells to generate antibodies against the antigens informations that is enveloped within the AAV vector. If you need to understand more about these approaches in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.